Okay, can somebody please confirm you can hear me?
Loud and clear.
Great. Let me share my screen here. My computer is still a little slow, so making the lecture file, so bear with me. All right, you should be seeing my screen. Yeah, we're 90% done now. All right, today is July 24th, and we should be discussing Lab 5, Streaking for Isolation and Culture Media. Also note that you need to read the unknown project file on Canvas for next week. I'll be talking about the unknown project next week, and it's best if you have the unknown project instructions read. Let me show you where that is. Close that down. Oops, didn't mean to do that. Uh, just close that down, not the stop the chair. Um, Now let me share my screen again. All right, should be seeing my screen again. I tried to shut down a file and instead it shut down the sharing. So next week will be week 5. Wow. So scroll down to week 5 lab. The unknown instructions, you need to read this file. I'll talk about the other links here, but this file you should at least look over, if not read it, before I talk about it. Any question about any of that? And good, the lecture file just finished, so my computer will start working better. So let's talk about Lab 5, Streaking for Colony Isolation and Culture Media. Can I shut this down? I guess I can. Lab 5, Cultivation of Bacteria, Culture Media, Inoculation, and Isolation. As always, there's some reading to do before you do the lab. So read about culture media, just a few pages. And then read about obtaining a pure culture, page 162 to 163. There are some video clips in today's lab. There is no virtual lab. Learning objectives for this lab. Oops, a little too far. Understand the importance of proper specimen collection and transport to the laboratory. Explain the 5 principal steps used by microbiologists to process specimens. Be able to differentiate between the different types of culture media, explain the purpose of streak plate culture technique, that's AKA streaking for colony isolation. And then 5, define the following terms: selective media, differential media, and reducing media. Any questions about any of this stuff so far? Get a little closer. All right. The ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation. They can dry out, and some are sensitive to the exposure to oxygen, all anaerobes, and therefore require a special transport medium to keep the specimen alive. Once a specimen is received in the lab for microbiological culture, you use the 5 principal I steps to process it. There's inoculation, isolation, incubation, inspection, and identification. Inoculation is just placing the specimen on an artificial culture medium to initiate growth. Isolation is spatially separating microbial growth into distinct and separate colonies. Incubation is simply placing the culture medium in an environmental condition to facilitate its growth. Inspection is examining the culture for the presence of microbial growth, and then identification, performing specific tests to ID any pathogenic microbes isolated. Any question about any of that? These 5 steps will be described in detail after we discuss different types of culture media. You're already familiar with general purpose media, NA, NA, TSA. These are media that contain ingredients to grow many different specimens, but they do not grow all specimens, and they're non-selective in nature. To promote the growth of some organisms, you need a special enriched media, and that would be to grow a fastidious microbe, then one that has a very specific nutritional requirement. For example, the bacteria that causes strep throat, Streptococcus pyogenes, is a fastidious microbe. It needs an enriched general purpose media to grow. And usually we grow this on red blood, not red blood cell, blood agar that contains 5% sheep's blood to provide the ingredients that Streptococcus pyogenes requires in order to grow, meaning the red blood cells are supplying the nutrients that Streptococcus pyogenes requires before it will grow. So that's an enriched media. Any questions about that? You use enriched media to grow a fastidious microbe. We also have selective
I
media.
had a question.
Yeah,
So enriched general purpose media, is that still non-selective media?
yes.
Okay, thank you.
We also have selective media that contain chemicals that inhibit or halt the growth of some microorganisms, but allow other microbes to grow. Keep in mind when you're using a selective media that the selective media isn't necessarily perfectly selective, meaning given enough time, an organism which is selected against may grow on it. And then second, even those organisms that are supposed to grow on the selective media may grow with smaller colonies or grow slower than they would on a general purpose media. An example of a selective media would be something like 7% sodium chloride media. It would select against microorganisms that would not grow in 7% sodium chloride because it has 7% sodium chloride in it, and would select for organisms that could tolerate 7% sodium chloride. An example of one that would grow on 7% sodium chloride would be all of the staphylococcal species like Staphylococcus epidermidis, Staphylococcus aureus. Those can grow on 7% sodium chloride media, which is a selective media. And then an example of one that would not grow would be Streptococcus pyogenes. It would not grow on 7% sodium chloride, as well as the fact that you're not adding the additives for that one. But something like E. coli should not grow on 7% sodium chloride media because it's selected against. We also have differential media. These media contain chemicals that produce visible differences among different group of organisms when they're grown on the media. The differences are unrelated to how well the organism can grow on the media. Usually, the colonies or the cells will appear different when grown on the media. So the dissimilar bacteria can be visually distinguished from each other. An example of a differential media is, what would be a good one? Well, this isn't the best example, but EMB is differential media. Trying to think of one that's only differential. Blood agar can be differential, but it's also enriched. I can't think of one that's only differential, at least at the moment. But EMB, when you grow your cells on EMB, they will show differences on their ability to ferment the sugar lactose. If they can ferment the sugar lactose, they will either be dark or metallic looking. And if they cannot ferment the sugar lactose, they will be their normal color or lack of color. Like if you grow E. coli on EMB, of course, that one can ferment the sugar lactose. If you were to grow Pseudomonas aeruginosa on EMB, it would be its normal color. It would not be turning dark or blackish, or would not be metallic in color because it cannot ferment the sugar lactose. Lastly, we have reducing media. This promotes the growth of anaerobic bacteria, which need low or no oxygen conditions in order to grow. Reducing media usually has something in it that removes the oxygen from the media, like thioglycolate media. It removes the oxygen from the tube, and that is a reducing media. It's important to note the culture media can be classified into more than one category. For example, excuse me. Usually I sneeze twice. Hopefully I won't sneeze again. MacConkey's is both selective and differential. Blood agar is both enriched and differential. Any question about any of that? All right, so MacConkey's agar is a selective media. If it grows on the plate, it's likely to be gram-negative because it selects against gram-positive cells. And gram-positives usually don't grow on MacConkey's agar. It's also differential, allowing you to differentiate between cells that can cells that can metabolize lactose and cells that cannot metabolize lactose. If it can ferment lactose, the colonies will appear pinkish, as well as the media around the cells will appear pinkish. So this cell here is gram-negative because it can grow on the MacConkey's agar, and it can ferment the sugar lactose because it's pinkish. This one here is gram-negative because it can grow on the MacConkey's agar, but it is not pinkish, so it cannot ferment the sugar lactose. And usually the bacteria will be their normal color, and that one is either white or off-white. Any question about that? This is blood agar. It is both an enriched media and a differential media. It's enriched because it can grow bacteria that will not grow on a general purpose media. You need to add an additive in order to get this fastidious organism to grow. And what we do is we just add the red blood cells, and then the red blood cells can provide the additives that some bacteria species need, like Streptococcus pyogenes. It needs an additive, actually two additives, and the red blood cells supply the additive. And so if this would be Streptococcus pyogenes growing here, it's growing on the blood agar because the red blood cells are adding the additive that Streptococcus pyogenes requires in order to grow. So it is enriched. It's also differential. It allows you to differentiate between the hemolytic pattern that the species can accomplish, meaning Streptococcus pyogenes can completely lyse the red blood cells while we're seeing this clear region around the white region, which is the bacteria growing there, and in between the beta, meaning the beta is the streak of bacteria, and this is called beta hemolysis because it's lysing completely all the red blood cells. Why it looks clear. You can actually show this up to a light, and you can see the light bulb. You can see through the clear region, not through the bacteria, not through the area where there's blood, but where it's clear, where the hemolysis is complete. And complete hemolysis is called beta hemolysis. Any question about that? You also have alpha hemolysis, which shows a darkening or a greenish tint of the blood. Alpha hemolysis is where you have partial lysis of the red blood cells, but not complete lysis. Let me blow this up. As you can see, there's red from the red blood cells here. And then right before the off-white alpha streak, which is the bacteria, we have a darkening. That darkening is from the partial hemolytic activity of this bacteria. And inside the alpha, it's also dark, and that region is showing the darkening better than actually this region here. And that's alpha hemolysis, where the red blood cells are partially lysed. This one is showing no lysis, where the red blood cells are red all the way up to the streak of the gamma. That's actually a gamma. And this we call gamma hemolysis or no hemolysis. And that's why that's a gamma, because this is gamma hemolysis. This is an alpha, because that's alpha hemolysis. And that's a beta, because that's beta hemolysis. Any question about any of that? So blood agar is both enriched and allows you to differentiate upon the bacteria's ability to engage in hemolysis. And you can see alpha hemolysis, gamma hemolysis, which is no hemolysis, or alpha hemolysis, which is partial hemolysis, and then beta hemolysis, which is complete hemolysis. Oh, let me state that normally around beta hemolysis, you do have a ring of alpha hemolysis, darkening of the blood cells before you get the beta hemolysis. So this table shows you the different media. We have general purpose media. There's 3 of them we're going to talk about in this class. TSA or tryptic soy agar, Na or nutrient agar, or NAY, N-A-Y-E, nutrient agar with yeast extract added. These are general purpose media. They promote the growth of most organisms. You cannot grow fastidious organisms on general purpose. They're non-selective. So both gram-positive and gram-negative can grow. And then there's a blood agar. It is an enriched medium, so you can grow some fastidious organisms like most of the Streptococcus species. And it's just the red blood cells make it an enriched medium. It's also differential because you can use the blood agar to differentiate among the species hemolytic pattern, whether it's beta hemolysis, alpha hemolysis, or gamma hemolysis. So enriched and differential. There's also chocolate agar or CAP, and this is an enriched media promoting the growth of some bacteria like Streptococcus pyogenes can grow on chocolate agar. Haemophilus influenzae can grow on chocolate agar. We also have McConkey's agar, which is selective and differential, and McConkey's EMB and HE are all selective and differential. They are all selective for growing gram negatives, meaning they select against gram positives, but gram negatives tend to grow on them. And they also are differential. You can differentiate based upon their lactose fermentation. If they engage in lactose fermentation, there will be one color. And if they cannot ferment lactose, they will be their normal color. McConkey's, if it ferments lactose, it'll be pink. EMB, eosine methylene blue, will be either dark color or metallic color if it can ferment lactose. Hectone enteric agar, if it can ferment the sugar lactose, it will be salmon colored, I believe, a yellow-orange color. And if it cannot ferment the sugar lactose, it will be the bacteria's normal color, which could be off-white, white, or some pigmentation. There's also PEA, phenyl ethyl alcohol. This is a selective medium. It selects against gram negative, so it's selecting for gram positive bacteria. And if it grows on PEA, it's likely to be gram positive bacteria. The PEA, the phenyl ethyl alcohol, gets in the cell wall of the gram negative cell and does not allow it to grow. And then there's mannitol salt agar. It's also selective and differential. It's selective because it has salt in it, 7% sodium chloride, which does not allow many species to grow. And if they can grow, then they can tolerate growing in 7% sodium chloride. It's also differential on the base of its ability to ferment the sugar mannitol. If it can ferment the sugar mannitol, it'll turn yellow and have a yellow halo around the bacteria colony. If it cannot ferment the sugar mannitol, then the agar will be its normal color around the colony. It will not be yellow. So there will be no yellow halo. And it's easy to know what's in mannitol salt agar. It's just the sugar mannitol and then sodium chloride, the salt. Any question about any of that?
Will we need to know what can grow in each type of medium?
You should know the different types of media, and you should know that McConkey's grows. You should know that McConkey's grows gram negative and PEA grows gram positive. You probably do not need to know, well, at this point, you don't need to know what is in McConkey's. In fact, I haven't told you what's in McConkey's media, but you don't need to know, for example, that E. coli specifically will grow on McConkey's media. You don't need to know that. But you should know gram negatives will grow on McConkey's media.
Okay.
Okay?
Perfect.
At least for now. Let me word it that way. All right. So inoculation is just getting a laboratory specimen from a patient and inoculating a dish to get the bacteria to grow. This looks a little bit mistaken. That shouldn't be there. It should be up there. There we go. And then isolation, you need to isolate the growth and get a pure culture so that you can study it. Like, for example, if you had a multi-mixture, like all these different cells, and then you ran a test and the test became positive, you wouldn't know which species was positive and which species was negative in the test. So you have to isolate the colonies and get a pure culture. Louis Pasteur developed the concept of working with a pure culture. It was a very difficult way of getting a pure culture, and he used limiting dilution of a liquid culture, and he made many different dilutions and then replica plated it. And if ever he got one tube out of 10 tubes that had growth in it and 9 tubes had no growth in it, he correctly concluded that the one tube had a pure culture in it. Robert Koch developed a much easier method for developing a pure culture, and that is he streak plated the cells on a solid media. And when he streaked it out the first time, he got isolated colonies. Each colony would only be about 70% pure, which is not good enough in microbiology. But if you take an isolated colony from the first plate and then streak it out on a second plate, once again to get isolated colonies, and then you take the cells from the second plate that had an isolated colony, the cells have now gone through 2 colony isolation events, and the chance of that being a pure culture is greater than 95%. And that is good enough for microbiology. And that we call streaking for isolation or streaking for colony isolation. And to do it, you need to streak it out on 2 separate plates, taking from the first plate an isolated colony to do the second streak on the second plate. And the idea is to get a pure culture, and then you can study it. So what you do to get a pure culture is get your mixed culture and then streak it out. Here we're showing you the 4-quadrant method, where you streak out in the first quadrant, and then you sterilize your loop, and you streak out the second quadrant. This isn't quite shown correctly because what you're doing for the second quadrant is streaking here, where there's no growth, to cool the loop, and then go through the first quadrant where the bacteria numbers are low, and that would be in this region here. You don't want to go to this region here because here the bacteria are almost at their full dilution. They're hardly diluted at all here. And as you streak, you're going to dilute out the cells on the loop so that this last one here will be the most dilute of the first quadrant. So you want to pick up from the low regions here, like I said, this region here, and streak in quadrant 2 starting here, picking up the cells in quadrant 1, and then streaking out quadrant 2. Any questions about any of that? Then you sterilize your loop again. I'm showing you with the mouse. Put it in the flame or the Bacto incinerator, and then start streaking out quadrant 3. This is a little better. It's actually showing you. You start streaking on the auger in quadrant 3, where there is no cells from quadrant 2, to cool the loop. And then you come into this region of quadrant 2 to get the bacteria that are diluted in quadrant 2. You do not come back into this region. So this is actually wrong, showing you here. You shouldn't go all the way up to here. You should stop around there and then streak out quadrant 3. And you should come to quadrant 2 to pick up cells about 3 or 4 times. We're doing that a little too many times here because that looks like 1, 2, 3, 4, 5, 6 times. You only want to do it about 3 or 4 times. And then you streak out quadrant 3, you sterilize your loop, and actually in quadrant 4, they've done it correctly. You cool the loop, and then you streak it in quadrant 3 where the bacteria numbers are diluted, and that would be this region here, not this region here. And then you streak it out about 20 to 30 times in each quadrant. It's not showing you 20 to 30 quadrant streaks, but if you look there, that's about 20 to 30 streaks. And there is actually probably more. And the idea is someplace along in your streaking, you put down isolated cells which will grow into isolated colonies. It doesn't matter if it's in quadrant 4, quadrant 3, quadrant 2, or even quadrant 1. If you had very low dilute cells initially, you might get your isolated colonies growing in quadrant 1. Usually it's not quadrant 1, it's some later quadrant. Any question about any of that?
I had a question.
Uh-huh.
So is the quadrant method the only like pattern or way to do the streak plate technique?
No, you usually
Okay.
use either the quadrant method or what is it, the 3-sector method? I don't remember what that's called. Trisector maybe? I don't know. Usually you do 4 quadrants or 3 sectors. Okay. And I've never seen 2, and you don't have to do even 3. You could do only 2, or you might even do only 1. For example, if your cells were low enough to begin with, you might get isolated colonies on the first quadrant. So you only need to do 1 on the entire plate, and that would get you isolated colonies. The idea is to get an isolated colony, and to help you get isolated colonies, we use either the sector method, which is 3, or the quadrant method, which is 4.
Okay, thank you.
All right. Oh, let me go back and state, once you get an isolated colony, remember that only about 70% of them are pure. To get a pure culture, you need to streak it out on take from this isolated colony, and then streak it out on a second plate, once again to get isolated colonies, using either the quadrant method or the sector method, the 3-sector method. And then you take cells from an isolated colony on the second plate. And even then, it's not guaranteed to be pure. You won't get 100% of the colonies to be pure, but it'll be greater than 95% chance that that colony will be pure. And that's good enough in microbiology. All right. So while you're doing your growth, you got to incubate the culture, and you just put it in an environment to help grow the bacteria. Usually we put our cultures in an incubator, and incubators can be fancy like this one, which it can control the temperature, it can control the humidity, it can control, if you look down there, those knobs are knobs controlling how much oxygen and how much carbon dioxide the cells will be exposed to. You have to shut the door, obviously, to get the oxygen and carbon dioxide levels are correct. And you have to shut the door to get the humidity correct, as well as, I guess you have to shut the door even to get the temperature correct, but at least temperature won't change that much. Most of the incubators we have at Clark College only control temperature, humidity, and then the time you grow the cells for. We don't have complex ones that control the oxygen and the carbon dioxide. Any question about an incubator and growing the microbes? The time is just how long you let it grow in the incubator. The humidity, what you do is you see that dish there? You add water to that dish, and that makes the air inside the incubator humid. That's important because if you do not add water to the dish, then the agar plates will only last about a week in the incubator. And then they'll start drying out, and you won't have viable cells after about a week. If you have this beaker or whatever dish of water, it'll make this humid, and then the agar plates will last at least 2 weeks in the incubator because the water does not dry as fast from the plate. Any question about any of that? And that's true of a tube. Most of the tubes are open to the outside, so the water will evaporate from the tube as well. I'm not sure what happened here. That shouldn't be there. That should be up here. Oh, I got 2 of those. Why do I have 2 of those? Let's get rid of that. All right, then there's inspection. This is looking at obvious visible colony morphological variations to start to begin to differentiate the species. Microbiologists can describe the colonies by a variety of colony descriptions, like their size, their shape, their margin, their elevation, and their pigmentation. That will not be enough to identify the species, but it begins to start to identify the species. And then you do identification where you run specific tests to begin to identify the species. Usually we do this by biochemical testing, but you could identify the species by sequencing the DNA of the species. We'll talk about the chemical tests we'll run, at least in this lab. All right, know the terms of this lab. You don't need to look at the references, but we have a little video for you to watch on inoculation of an agar plate using the streak plate culture technique, meaning streaking for colony isolation. You only need to watch that movie to 4 minutes and 15 seconds. Note that this movie or video is using the 3-sector method, not the quadrant method. And then we have a little video on showing you beta hemolysis, alpha hemolysis, and gamma hemolysis. So watch those. And then watch a little movie on growing bacteria on MacConkey's agar. For this movie, you only need to watch it for 3 minutes and 50 seconds, and then you don't need to watch it any further. And then we have a little movie on growing bacteria on a mannitol salt agar plate. And we do not have a virtual lab anymore for this one. And then begin working on the worksheet, answering the questions of the worksheet. Let me blow this up a little. Describe a natural circumstance where Staphylococcus species of bacteria might benefit from tolerating salt. So you can't say growing it on a 7% sodium chloride media plate, because that's not a natural circumstance. Where can you find a Staphylococcus species growing naturally with tolerating salt? And then 2, how might hemolysis benefit a hemolytic organism? So if we're growing a hemolytic species on like blood agar, what would be the benefit to that species of hemolyzing the red blood cells. And then 3, imagine you have a culture medium containing a single bacterial species. The species may be one of several possible species. Describe a scenario where you use mannitol salt agar, blood agar, and EMB plates to identify the unknown bacterial species. You need to name 2 properties to each of these plates to help you identify the species. Any question about number 3? And if you don't name 2 properties to each one of these 3 plates, you will not get 100% correct. In fact, for this question, if you only name 1 for each 3, you only get 50% correct. Any question about number 3? All right, number 4, what's the main goal for streaking for colony isolation? That is stated in this lab. And number 5, what happens to the growth on the plate if you do everything correctly, but you forget to sterilize the loop in between sectors when streaking for colony isolation? You need to be specific, discussing the growth on each sector. So let's say we have 3 sectors on a plate, and you are doing it correctly, so you are sterilizing the loop before you begin sector 1. And then you streak out sector 1. And now you're not doing it correctly, and you don't sterilize the loop between sector 1 and sector 2. And then you streak out sector 2. And then once again, you don't sterilize the loop in between sector 2 and sector 3. When you streak out sector 3, so be specific, discussing the growth on each sector, and tell me what's going to happen. And this is a thinking question. It's not an easy question, but usually most students get it correct on the second try. So many students will miss it on the first try, but once you work on the second try, most students do get this question correct. Any question about any of that? All right, number 6, also a thinking question. What happens to the growth on the plate if you do everything correctly, but you transfer from stock culture to inoculate each of the sectors? You need to be specific, discussing the growth on each sector. So you're sterilizing the loop, cooling it, and then picking up the cells from the stock culture, and then streaking out sector 1. And then you sterilize the loop, and then you cool the loop, pick up cells from the stock culture, and then streak out sector 2. And then you do that again for sector 3. This once again is a thinking question, although students are more likely to get number 6 correct the first time than number 5, but maybe only about half of you will get it correct the first time. Most students do get this question correct on the second try. Any questions about 5 and 6? And by the way, the growth pattern will differ from 6 and 5. Number 7, suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species, meaning you started from a mixed culture, and you streaked it out one time on a plate using colony isolation, so you're doing either 3 sectors or 4, and you got isolated colonies. How can you use this plate to create a new pure culture of only one of the bacteria? You must be specific in your answer. Any question about number 7? Number 8. In the table below, we have a variety of agar media available to microbiologists to cultivate bacteria. Denote how many media, denote whether each medium is selective, non-selective, or differential by placing an X in the appropriate column or columns. You may put more than one X in the table for each row. You don't have to, but you may. You may use the table of media in the lab module to help answer this question. So nutrient agar, if you think it's selected, put an X there. If you think it's non-selected, put an X there. If you think it's differential, put an X there. And if you think it is two of them, put two X's in. I'm going to give you a hint here. We never have a media which is both selective and non-selective. And do that for NA, TSA, do that for CAP, do that for BAP, MAC, EMB, HE, MSA, and then PEA. Any questions about number 8? For number 9, we have 3 different plates here. Which of the agar plates or plates were inoculated without using the streak plate technique? If you think it was A, answer A. If you think it was B, answer B. If you think it's C, answer C. If it's 2 of them, put 2 answers. Any question about any of that? All right, that's it for lab 5. Go ahead and work on the lab. I will be here until 8:30 to answer any questions with the lab, but answer them before all the students log off, because once all the students have logged off, I too will log off. Any questions before we get started? Somebody had started to ask a question in the lecture and I didn't understand the question. If you want to re-ask your question, you can do that now. All right, I'm not hearing anything, so get started on the lab. I'll be here until 8:30 or the last student logs out to help you with the lab.